So this is a quick video on something we should appreciate or learn more about in the hobby. So this is something not a lot of people talk about, and this is the topic of biofilms. Biofilms and bacteria. And sadly, this is something I never really managed to study much at university. I am awful at biochem, and bacteria was something that kind of got shoved in the biochem box. So that was medicine sort of things, and like I appreciate their ecological and physiological sort of biology. Their biochem, no. So why should we appreciate them? Firstly, they're in our filters. That is the major thing in our filter that keeps this hobby working almost. Some fish maybe not, but also for many fish this is a food source. So bacteria are what they eat. Balancistes xanthellus is my favourite example. But also Rift Valley cichlids, they will eat biofilms. They eat ulfrich, and biofilms are a major part of ulfrich. So I will write a blog post for this, but since I've only just come up with the idea of the video, it might take a while. So what are biofilms? Biofilms are bacterial colonies. They exist in a sort of matrix which contains, my terminology isn't going to be great, I haven't researched this in a while, but sort of networks of tunnels. And it's not just bacteria that live in these tunnels. You might notice biofilms have a sort of colour stain, and the bacteria, there are many different types of bacteria, and there are photosynthetic bacteria such as cyanobacteria. So it can have a colour due to the bacteria in it. So this you often see mostly clear ones. And do biofilms just inhabit aquariums? No, they are in your mouth, especially a lot of mouth decay is due to certain bacteria in your mouth. They are in your gut, they are everywhere. But biofilms can be, they exist in this matrix, and it can be anywhere from really thin to be quite large, wispy. There's so many different types of bacteria. So it's not just bacteria actually inhabiting these. The bacteria make it, but then also within it is protozoa. So, and now I've forgotten any name of protozoa. Chlorates? No, but a lot of protozoa will inhabit these, and they will sort of sit in the sort of tunnels, collect, because the biofilm will sort of shove water flowing through, or shove not just water, it depends on where the biofilm is, but will sort of shove oxygen and nutrients through the biofilm. And this means that the protozoa love it, and it's great for them. And that can also give it a stain too, because protozoa can be photosynthetic, so that's where you get green ones and they get confused with algae. And it's the same with bacteria. And then algae will also inhabit it. So algae are not bacteria and they're not plants. Seaweeds are not plants, they are algae. They are macrophytic algae, large algae. But I am talking about more single-celled algae. I guess you could get multiple-celled algae within biofilms. You might also get amoebas and all sorts. So they are brilliant, and they do feed off nutrients. This makes them a brilliant food resource, and a lot of things will eat them. Generally within the sort of freshwater environment, they are very thin, unless it's in a slower sort of moving area. Along rivers they are thin because any higher and they're just top layers will get washed off. So should we appreciate them? Yes, they are amazing structures. People say corals are amazing, and corals I don't think are that cool compared to biofilms. And why are biofilms cool? Because they have this ability to talk, each cell can talk to each other. Corals, I guess they have chemical signals, But back to biofilms, each cell can do what's called quorum sensing. So it can send signals to each other, and it's almost, they're amazing for something that is single-celled. A biofilm is a colony of single-celled bacteria. Usually, I think all bacteria are single-celled. That's going to sound really stupid, isn't it? But this is like, they're absolutely amazing structures, and a lot of people hate them. People hate anything, I swear, that's in their aquarium that isn't fish, plants, or the decor and the substrate. If they see snails, they just eradicate them. But a hydra as well. These are all parts of the natural ecosystem in fish. A natural aquarium, how is it natural if it doesn't include what's naturally in the ecosystems themselves? Firstly, half the aquarium plants aren't natural. This is not natural because why would you, half these aquarium plants are even riparian vegetation, so they're sort of just above the waterline and they like high humidity, so they can kind of cope underwater. Or they're just not aquatic at all. You do get aquatic plants, balis, but you'll notice aquatic plants tend to, when out of the water, they just flop. So yeah, water lilies, obviously, floating plants, yeah, but we should enjoy biofilms. They are beautiful, and especially in the aquarium, they are a food source to many other animals. I always get the odd biofilm. I hardly know where they're coming from. I encourage them on my sponge filter. So my sponge filter, I will clean, I'll just squish, but it is only there to grow biofilms. So my ancestors particularly can feed on it. I've never noticed my brown ancestors, Xanthelas, feed on the sponge filters, because the filters act as a good place for flow. So they will take a large amount of water through and up. So that makes them really good structures for actually sort of growing these films and for feeding the fish of their form. Well, it's sort of like not as a food, but as a sort of additional natural thing. So I never rely on it. In the filter, your filter is full of biofilms, and this is why there is no water filtering bacteria that were nitrifying bacteria in the water column, because they're in biofilms. Biofilms don't float around. They attach to hard surfaces, because if you're floating around, you're just going to get bits knocked off, a lot of abrasion from the water. So they live in the filter, on the sponges itself. It is, well, not just sponges, ceramic media, but it's a good idea to clear the waste from them so they get more flow. And when you dechlorinate your water, you're not making it safe for the fish, really. Fish can withstand quite a bit of chlorine. You'll make it safe for the actual bacteria within the filter. So it's kind of a misconception, and that's probably why a lot of people actually clean their filter media under the tap, which is a horrible idea. But you're told in the store that chlorine is bad for the fish. You're never told anything about bacteria apart from it needs to build up or the filter needs to start working, which is like, no, the bacteria need to grow. They're a colony. Think ecology. So it's important to respect and understand what you're actually doing. Bacteria are specific. They have what's called a range of tolerance. So your bacteria will change as temperature changes. I doubt they'd quickly die off. One of the most important things to remember about bacteria is, well, basically the laws of physics. Nothing can survive without the food to feed it. The food, the oxygen. So you're not going to have excess bacteria. There's only ever going to be enough bacteria to feed whatever, there's only ever going to be enough bacteria in the filter, well, filter's down there, to deal with the waste from however many fish you have in there. So you take away a portion of the filter media, and what's that going to do? Well, if you take away a portion of the filter media, you're removing an unknown portion of the bacteria, because you can't see the bacteria. You need an electron microscope. These are the more than £1 million microscopes to see bacteria, because light microscopes don't go that small. Because bacteria is smaller than an animal cell and even plant cells. So you have to consider that you don't know how much is in each part of the sponge. You can kind of do an educated guess that the regions with the highest flow are going to have more bacteria, regardless of how much space, how porous they are. How porous will just indicate how much flow can go through. But then sponges are porous, and the only issue is the more porous the smaller the holes, the more easily they get blocked. So there's a lot to consider. And when people were like, I want to change my filter, I will run them together. Well, you run them together, and then when you take the old one away, the bacteria doesn't think, oh, she wants me to go into that filter. No, what's gonna happen is the bacteria is gonna there will be bacteria in the new filter and in the old filter. Whichever is more suitable for the bacteria will have a higher number of bacteria in it, but the other one will still have bacteria in it. So it's kind of like proportional. If you think population ecology, so there will only be enough individuals as the food that is there. So a filter which supplies more food and more oxygen will have more. So then you don't know how much you're taking away. If your filter is, say, 2/3 higher turnover, yes, it will contain probably around 2/3 more bacteria after amount of time. But you're still removing 1/3 of bacteria, and that's a mini cycle. And this is how people result in uncycled tanks. It's not easy this is the kind of stuff you really have to think about, and it's worth considering because you don't want to make the mistake of almost uncycling your tank. So what would I do? Just move all the old bacteria into the new filter and remove most into the new filter and slowly change over the media if you want to whatever whatever media you want to put in. Do not change that at all. You are removing all the bacteria. It doesn't just live in there in the soup either. It is attached to the actual media itself. And I have a thing about media. I like sponges. You can clear the waste off them. You can actually give it flow you can squish it, the waste comes out. With ceramic media, you squish it, you can't even squish it, you crumble it. So removing the actual waste particles is quite difficult, especially from within. So that means you actually have to almost throw away ceramic media. I think you could, environmentally friendly, but the bacteria is going to grow in a biofilm. So no matter the surface area you give it, it won't change the amount of bacteria that will be on the actual media itself, because it will just build up on top of each other. And bacteria is tiny, really. So I do have ceramic media, but I don't like to throw away stuff if it's of use. Obviously there's some stuff that isn't out of date fish food, especially if it's a year out of date and I've forgotten that it exists. So also chemical filtration, I'd never use it. I just have biological and mechanical. But mechanical, you have to remember that you want some mechanical filtration, as in floss, wet dry pre-filter, something like that, so to maintain to clear waste before it reaches the actual bacteria, so it doesn't clog it, so the bacteria still remains getting a lot of flow. So I've probably ranted a lot, and I can't explain it very well, but a lot of people do forget the bacteria that is there. Biofilms is the word, and it's worth considering because it will stop you making a lot of mistakes. It was also worth remembering that bacteria without the flow and without the oxygen will die, but it can take a while. I usually say 2 hours is when you start to get this sort of drop off, but it can go into a dormant state. Changing the temperature will mean that different bacteria, you kind of get a succession, so some bacteria will be lower numbers because there's plenty of species, and then as you change temperature, some will increase, others will decrease. Same with pH, salinity. So if you do things gradually, almost changing perimeters, it makes things a lot more stable. I've never noticed any difference, but I prefer to change things gradually because not just the fish, but the bacteria, because there's so many species. And don't fear the bacteria. It's not the bacteria that's going to harm you. I would talk about disease, pathogenic bacteria to humans, but I haven't done enough research in it, and I'd like to get some reference photos before I even talk about it. And yeah, just treat your aquarium like an ecosystem. You can have natural stuff, you can have unnatural stuff, but it's fine to have biofilms growing. This wood loves to grow a bit of biofilm, there's a bit there. And it's fine to have hydra. This tank has freshwater limpets from Tubiflex. The other one has freshwater hydra from Tubiflex. So Tubiflex brought in some stuff. It also had a random worm. I think this actually has live Tubiflex under the substrate, but just leave it be, it's an ecosystem. It's natural to have a variety of different organisms in there. So I do have the snails, they're a pain, produce a lot of waste, but it's just more natural to have, and it feeds the fish because, well, if you have Lorocarde, they like to rasp, so why not give them a few other things to rasp on if they're going to take it? And just a stable aquarium shouldn't really be growing certain algae on higher levels. Unless you're aquascape, you could get some really odd, beautiful algae growing. Anyway, thank you for watching. I'm happy to answer any questions, or it'd be interesting to hear anyone who does microbiology, their opinions on the topic. I'd also like to see, I can also provide any papers give people links to papers if they don't really know where to start looking. Anyway, thank you for watching.